So I want to start with a really high-level introduction into why working on human genetics is so awesome. And the reason human genetics is valuable is because it provides us with a lever that allows us to move between two different kinds of information, both of which we've become very good at generating over the last couple of decades. So one of those classes of information, of course, is information about the function of particular bases in the human genome. In this picture here, we have a track from the UCSC Genome Browser that shows one sort of little slice of information about the human genome, providing information, for instance, about which chunks of the genome are actually expressed and in which tissues, and which sections play key regulatory roles, and again, in which tissues. And that type of information continues to grow thanks to the efforts of projects like ENCODE and Epigenomics Roadmap and GTEx and others. Another type of information that we've actually already heard quite a bit about today, that we have become, as a community, very good at generating is phenotype data on hundreds of thousands of people. And this includes information on disease diagnoses, of course, on electronic medical records, prescription registers, and then a whole host of other types of information that we can now collect from human beings using both low tech as well as high throughput fancy genomic methods such as transcriptomics and epigenomics. And the power of human genetics is that it allows us to move between these two worlds. So it basically provides us with this transition point, and we can take, for instance, as we have done traditionally in human genetics, we can take people who have particular phenotypes. This might be a set of people who have a specific rare disease, or collections of people who have a common disease like type 2 diabetes, and we can use human genetics to determine which genetic variants those people have in common, and that will point us towards particular regions of the genome that are associated with that particular disease. And that, of course, is the standard kind of forward genetics approach to understanding human diseases. But as our data sets of human genetic data and phenotypic data grow, we can also start to move in the opposite direction, to do reverse genetics. Basically meaning we can find bits of the genome that we think are particularly interesting for some reason. We can then find those people in our big genetic databases who have genetic variants in those regions. So let's say, for instance, as Seik was talking about today, all the people who are knockouts for a particular gene. And then we can go into our biobanks and look to see what particular clinical features those people have in common. And this kind of reverse genetics has now become possible thanks to the advent of massive sequenced biobanks. And we live in a world of almost embarrassing riches when it comes to human genetic data. So the exact number is actually difficult to estimate, but it's very clear at least 1 million people around the world have now had either their whole genome sequenced or, more commonly, their exomes, that is, using a particular technology that captures just the protein coding regions of the genome. And this is an enormous amount of data that provides us with information about the patterns of genetic variation across the human population. In theory, at least, we could combine all of this data together to learn much more about the evolutionary history of humanity, as well, of course, as the genetic variants that underlie both common and rare disease risk. And this data grows exponentially. I'm not going to bore you with the same curves that you've seen in many other settings. I am just going to show you how quickly data grows in one place. This is data from the Broad Institute, contributed by Niall Lennon. It basically shows the growth in the number of genomes in blue and the number of exomes in red. This is cumulative numbers over the last several years. And you can see that in 2016, the Broad generated twice as many genomes as it had in all of the previous years combined. So the amount of genomic data that's currently being generated is enormous and continues to grow our ability to look at these patterns of variation. Now, the challenge, of course, is that for basically any individual researcher anywhere in the world can't access all of this information at once. And that's true for a whole host of different reasons. These data have been generated by many different groups, both academic and commercial groups. There are mundane obstacles to sharing data. It's difficult, still actually surprisingly difficult, to move data from one place to another, although this is becoming less of a problem as we start moving towards cloud-based solutions. There are continuing regulatory challenges with amalgamating DNA. That is cumulative data, that's right. That is the number on the x-axis. So you can see the Broad has now generated, nearly 70,000 whole genomes and about 250,000 exomes. So there are continuing regulatory issues with data sharing. The samples that have contributed to many large-scale sequencing projects were collected over decades. So that means that some people were consented before genome sequencing existed, and certainly well before the concept of open genomic data sharing existed. So that means they often were not consented appropriately to allow their data to be used in large-scale data sharing exercises. There are objections to data sharing. Large amounts of data in the genomic space have been generated by commercial entities who view that data as being valuable for their financial success. You may be shocked to learn there are even some academic groups that don't believe in widespread data sharing. And then another pernicious problem that plagues this area is the fact that there is inconsistency in the pipelines that we use for processing genomic data and for calling variants. So that means if we were to naively take all of the end results of each of the different pipelines that people have used to generate these variant calls and then try to munge all of that data together into one big resource, what we would end up with is a data set that was dominated by technical differences between different centers rather than being able to zoom in on interesting biological differences. So to help to address some of these problems, about 3 years ago now, we formed, with a large number of investigators around the world, we formed a group called the Exome Aggregation Consortium. And then the name of the consortium provides pretty important clues about what we actually do. It's very simple, actually. We collect raw exome sequencing data, we aggregate it, and then as a consortium, we put all of that data through the same processing pipeline and variant calling pipeline, and then critically, we make the summary data available as quickly and as openly as possible to the wider community. And the major driver behind this effort was really to make sure that we had great reference frequency data available for those of us who are interested in looking at the variants that we find in rare disease patients and understanding whether those variants are actually rare or not. So the first release of the EXOMAG data set, which we released back in October 2014, contained data from a whole range of different, mostly case control studies of complex diseases like diabetes and heart disease, include many of the samples actually that Saik talked about in his talk earlier today from studies of myocardial infarction. And we were able to release summary data for nearly 61,000 individuals, where we had restricted that data set to very high quality exome data from unrelated individuals, where the individuals had consent that was consistent with public data sharing. To the best of our ability, we removed individuals who had severe pediatric disease as well as their first degree relatives. The goal here was to make a resource that we could use as a kind of comparison reference for patients with rare diseases. So we wanted to make this more representative of the general population. And we put all of that data through the same pipeline and did joint variant calling across all of those samples. That data's been out there for a while, and I'm not going to go through it in detail. Those of you who are interested in still accessing it, it's archived at exact.broadinstitute.org. And because this provided such a large-scale data set, particularly for people studying rare diseases, it's been very heavily trafficked. So I think now is the most widely used reference data set of human genetic data with over 7 million page views. You can actually look at the plot of page views over time here. This shows us very clearly that human geneticists don't work on the weekends. You can see those little dips there. And there's basically no access at all of this data set at Christmas. So there we go. Okay, so EXAC is big and it's diverse. So here I'm comparing the size of the data set, and it's colored here by continental ancestry defined from genetic data compared to two previous publicly available data sets of human variation, the 1000 Genomes Project and NHLBI's Exome Sequencing Project data set. So it's large and diverse, and that gives us a lot of power to filter the variants that we find in our rare disease patients. So just to give you one example of how well this resource works, here what we're doing is comparing the power of ESP, the previously largest publicly available set of exomes, to EXAC for filtering variants that we find in individuals from 5 different continental ancestries. And the type of analysis we're doing here basically is saying how many variants are left if we throw away all the variants that are confidently over 0.1% frequency. So this might be the type of analysis you would do if you were looking at a patient who had a dominant disease, for instance. And what we find in this analysis very clearly is that compared to the previous data set, we see almost an order of magnitude reduction in the number of candidate variants we have when we filter with EXAC. And again, that's both because of its size as well as its ancestral diversity. So the key message from EXAC is that the more people we have from the more diverse backgrounds, the better we do, which I think is a message that the current administration could also stand to learn. So there are lots of scientific things that we can also do with a data set of this scale. And I think one of the most interesting, I'll just tell one story here, I could talk about more, but one story here is about data that, analyses that look not at the variants that are present in the data set, but variants that are missing from the data set. And this was work that was led by Caitlin Samoka, a graduate student in Mark Daly's lab who worked with us for the course of this experiment. What Caitlin did is to develop a clever little statistical model that allows her to predict for any gene or for any region of a gene how many variants should be present in that particular region under a random mutational model. This turns out to be very well calibrated. And then using that model, we can then say, so she can say how many variants should be present in 60,000 people if only random mutation was acting there. Then we can say how many variants are present in that region in the EXAC data set. And the difference between those two numbers tells us how strongly natural selection is actually acting on that region or that particular class of variation. And in this plot here, what you can see is the difference between Caitlin's model on the x-axis, that is the expected number of loss-of-function variants in each gene in the genome, and then the observed number on the y-axis, the actual number of loss-of-function or protein truncating variants that we observe in our 60,000 samples. You can see here that most genes fall below the line. That is, they have fewer loss-of-function variants than we would expect to see by chance. That's no surprise at all. What it tells us is that in most cases, protein truncating variants do something bad and they get removed from the population by natural selection. What's cool about this data is that for the first time, we could actually calculate how bad those loss-of-function variants are. In other words, how many of those variants are missing. Just to show you how useful that is, here is a gene where we know exactly what happens when you introduce either missense, that is amino acid protein changing, or loss-of-function variants into a gene. This is a gene called DINK1H1, where de novo heterozygous loss-of-function or missense variants cause very profound and early onset neurodevelopmental disorders. And this gene, you can see that the gene has almost exactly the number of synonymous variants that we would expect to see by chance. Synonymous variants are variants that don't change the amino acid sequence, so that suggests that our test is well calibrated here, but it is missing 2/3 of the expected number of missense variants, and basically all of the number of loss-of-function variants we would expect to see by chance here. Of the remaining 4 observed loss-of-function variants, 2 of them are artifacts, we can see, and there are actually probably 2 individuals in this data set who carry genuine heterozygous LOF variants and probably do actually have a neurodevelopmental phenotype. So that's for a gene where we know exactly what happens when you knock it out, when you have these loss-of-function variants. Here's a gene where we don't. This is UBR5, where really all we know is that if you knock it out in mice, it kills them before they're born. We don't know what the human loss-of-function phenotype is. We know it's involved in the ubiquitin pathway. But we can see very clearly from this data that it's missing almost half of the expected missense variants and basically all of the expected loss-of-function variants. So why we would put some serious money on this gene, on heterozygous loss-of-function variants in this gene, having some relatively profound disease-causing phenotype. We just have no idea what that is. And of course, one possibility is that these heterozygous LOF mutations kill people before they're born, and that's why we haven't yet detected that disease. So in addition to being able to apply that method to whole genes, we can also apply it to individual regions. Here's just one case study that I'll show. This is a gene called CDKL5. We know that missense mutations in this gene cause severe infantile seizures. And if you were to look at all of the missense mutations that have been observed in epilepsy patients so far, you'll see that almost all of them cluster in this region of the gene up here, this well-annotated region of the coding sequence. And indeed, that is exactly the same region where we see a depletion of missense variants in our data set in the general population. So that tells us that even if we didn't know that this was the most important region in this gene based on the disease mutations, we would have known to prioritize that region based on the absence of such mutations in the general population. And that turns out to be true more generally. So overall, if you take genes where heterozygous mutations are known to cause severe disease, and you take all the mutations that are found in the ClinVar dataset, that is a dataset of known disease-causing mutations, 89% of those ClinVar missense mutations fall within the 14% most constrained sequence in the human genome. So this really does allow us to zoom in on regions of genes that are the most likely to actually contain disease-causing mutations. And that's already proving useful in the discovery of new disease regions in autism, schizophrenia, and severe dominant disease. Okay, so that's all stuff that we did with the EXOMAG dataset, which has been out now since 2014. EXOMAG is great. 60,000 people, exome sequence from 60,000 people is a lot, but it's still not big enough. And it's still not big enough for a whole bunch of different reasons. It's not large enough, for instance, although we can look for 2/3 of the genes in the genome, we can look for depletion of loss-of-function variation. For about 1/3 of genes, we still don't have statistical power because we don't have enough samples. Much of the data in the EXOMAG dataset is relatively old and generated on older exome capture platforms, so it's not as good as more recently generated exomes. And that's a challenge if you're doing really nice, high-quality exomes for your patients and comparing that data to a much older reference dataset. It's missing key populations, like the Ashkenazi Jewish population, for instance. And it's, of course, because this is based on exome data that is focused on protein coding regions only, it's missing all variation from non-coding bits of the genome. So we set about generating a new data set, which we finally released in made our first release of in October last year, and actually just made this week our release of the full data file. This is called the Genome Aggregation Database, or Nomad. It's, again, inappropriately capitalized just to confuse everyone. And this data set consists of just over 120,000 exomes. So we've doubled our number of exomes in this data set, as well as for the first time over just under 15,500 whole genomes, giving us our first real insight into the patterns of genetic variation in non-coding bits of the genome. The exomes and the genomes we called separately, but then we merged them together for our downstream analysis, and that's also the way that they're displayed on the browser. We've been able to include new populations here. So you can see this tiny little pink section here, which corresponds to more than 5,000 individuals of Ashkenazi Jewish descent, which of course is a really incredibly important population to have for those of us who work on rare disease cases that come from the Ashkenazi Jewish community. So like EXOMAG, the Nomad dataset relies entirely on the generosity of individual PIs who've contributed to this dataset. And in total now we have about 107 PIs who have allowed their data to be included in the Nomad dataset. Nomad itself does no sequencing. We are opportunistic data parasites. We take advantage of data that gets generated primarily for analyses in the types of studies like the ones that Sake talked about earlier. And in fact, Sake himself contributed about a third of the data in this dataset. So huge thanks to all of the PIs who allowed their data to be used in this resource. So now I'm not going to go in detail through the variant calling process that we applied to the Nomad dataset. It starts with a huge amount of data in the form of raw short read sequencing data. We then put it through a whole series of steps using publicly available code, the GATK code base. So we start with about 3 petabytes of sequence data. That's 3,000 terabytes, about 12,000 MacBook Airs worth of data. And then going through this process, that leaves us with about 40 terabytes of variant call files or VCFs, which is actually an annoyingly large amount of data that we then have to mine through to analyze and then release the summary statistics that go out to the public. So these are the raw numbers. These are huge thanks, I should note at this point, to the Broad Genomics platform, which generated 90% of the data that was used in this dataset, and also funded almost all of the Nomad project. We actually have very little funding, external funding for this project. And the Broad Data Sciences platform, led by Eric Banks, and including lots of people who are named here, was responsible for building all of the GATK infrastructure and also for running the variant calls that were required for this project to work. But I did also want to thank, in particular, 2 postdocs, Konrad Kaczewski and Laurent Franchioli, who led the analysis of the exomes and the genomes, respectively. Jessica Alfoldi went through what she calls the nightmarish hellscape of the regulatory approvals that were required to make this happen. And Monkul, Laura, and Kristen were heavily involved in the QC of this particular dataset. And I also wanted to give a plug for a particular toolkit that we would have been completely screwed if we didn't have access to. There's a wonderful toolkit called Hale. Again, it's been developed at the Broad by a software development group led by a guy implausibly called Cotton Seed. Hale is a heavily scalable genomic analysis toolkit. It can run on all sorts of different computational architecture. We ran it very heavily both on the cloud as well as on our own internal supercompute. It's super easy to use and also very reusable and fully open source. It's based on the Apache Spark infrastructure. So anyone here who is thinking about doing massively large-scale genomic datasets doesn't have to be on the scale of 150,000 samples. Even if you're looking at 10,000 or 5,000 or 1,000 samples, Hale is a phenomenal resource for going through the QC and analysis of those datasets. So once we had generated this huge dataset, we were able to start doing some science. This is one of the first plots that we generated across the Nomad dataset. This rather hypnotic rotating image represents the outcome of a principal component analysis, which basically shows us the major axes of variation across the genomic data that was generated here. Basically gives us a sense of the ancestry of the individuals who've contributed to the dataset. And you can see, again, as this rotates around and into view again, you can see a big swathe of European ancestry samples down here, but also, over here, but also a large and good representation of individuals from a whole range of other ancestry groups as well. And we then assigned ancestry using a random forest approach based on 40,000 samples of known ancestry. We discovered a lot of variants as a result of this project, the majority of which are actually new. So in total, we found 17 million variants in our exome data. 6 million of those are completely novel. They have not been reported previously in any dataset. This is the first time that we've started looking at genomes. We discovered 260 million variants in the genome dataset. Again, more than 50% of them novel. Lots of loss-of-function variants and 2,500 genes that are plausibly knocked out in at least one individual. And over 10,000 pathogenic variants discovered in this dataset that were missing from EXOMAG. Those of you who are interested in accessing the data, the Nomad browser is available at nomad.broadinstitute.org. Massive thanks to Matt Solomon and others for putting that whole data set together. And this allows you to look, for instance, at the coverage across your favorite gene of exomes shown here in blue and genomes shown here in green. And you can see here, for instance, for the ApoE gene, for this key region over here, which is associated with Alzheimer's, and of course we heard talk about this earlier today, you can see that this region is extremely well covered in the genome data. but not well covered in exome sequencing data, showing one of the advantages of the newer genome sequencing approaches to understanding some complicated regions of the genome. A quick word on restrictions on publication and usage. It's very easy. You can use it however you want. There's no embargo, there's no data restrictions, and this is testament, I think, again, to the generosity of the PIs who contribute to the consortium in allowing their data to be released to the community in a way that anyone can use in any way they want. We have a lot of things currently in the pipeline. The early papers that will be emerging from this dataset include a deep analysis of protein truncating variants, looking at the sensitivity of all of the genes in the genome to loss-of-function variation. Secondly, detecting constraint in non-coding regions of the genome, taking advantage of our genome data, and we'll be increasing the number of genomes we include in a second call set later this year. Improving methods for rare disease diagnosis, working with Gonzalo Abacassis, who presented at this meeting last year, to federate our browser with the TopMed dataset, which is another very large-scale collection of human genome data, and finally and importantly calling structural variants to access a particularly important class of variation. And I'll skip over this except just to say very briefly, we're also beginning pilot studies where we recontact the 40% of individuals in the recontactable components of Nomad and look to see, based on their genetic data, what sort of interesting phenotypes they have. There are lots of challenges left, and because I was told to talk about slightly controversial things here, I wanted to at least mention them. We can bring these up in the discussion as well later on. There are three areas that I think are hugely underdone at the moment in human genetics that we need to really think hard about, and a number of people are already thinking hard about, but we need to figure out how to make this work. We need to, as a research community, think very carefully about systematically consenting participants, both for broad research usage and for genotype-based recontact. It's shocking how many samples are still going into large-scale sequencing studies where the consent is actually extremely narrow, and that means they're not usable in other settings. We need to come up with explicit regulatory frameworks for genomic data aggregation to say it's okay to release aggregate data to the wider community. We're still in a situation where it's very uncertain, and where it's clear that it's okay to share summary statistics across data sets. And at the moment, we push data sets like Nomad out without necessarily being in a situation where it's explicitly regulatorily okay to do so. And finally, it would be lovely if we could somehow build a system for global sharing of genomic data that isn't fundamentally broken. And it is true, I think, as most people here in this room know, that the current NIH-supported system, dbGaP, is not capable of coping at the scale that we're currently producing data. We have a lot of work to do, and we as a research community should be really taking the initiative here. We have a lot of work to do to move towards a system that actually serves the needs of the research community. So with that, I will finish by thanking everyone who's been involved in generating this data set. I've mentioned most of the people here already, but again, all of the PIs involved in it as well, particularly as Conrad and Laurent and the Broad production team. And if people are interested in working on this data set, we have a number of jobs available. Thanks very much.